I am Professor Durairaj Sekar from Saveetha Institute of Medical and Technical Sciences, Chennai, India. So our group is working on microRNAs. So microRNAs is a short RNA. Everyone knows that. So it's a kind of RNA, but it's not coding for any protein. That's the beauty of this microRNA. So this is a non-coding RNA and regulate various gene function, especially tumor suppressor or oncogenes. So what it do is, in the cells, it originates from the nucleus and processed in the cytoplasm, where it activated, and then it go and bind to the corresponding target gene. So if it binds, what will be happen is, so it will go and degrade the target genes, and then the disease may be progressed or suppressed. So there are a lot of hypotheses is going on using anti-microRNA or microRNA mimics, that is equal to microRNA. So with the use of these two molecules, we can able to suppress these microRNA in order to save the cells. So let me going onto the slides, what it do and how it possible how this microRNA induce the disease progression, especially oral cancer. So as we know that these microRNAs are very short non-coding RNAs with the nucleotide of 18 to 22 nucleotide in length. And as I said, it plays various key role in the regulation of cellular functions, such as proliferation, apoptosis, and gene expressions. So what is circulatory microRNA? Circulatory microRNA is nothing but you can detect in the serum, saliva, semen, and all kind of body fluid. So it is circulating throughout the body. So we can easily detect and able to observe or see the expression in the cells. So maybe in other words, we may consider this circulatory microRNA as a non-invasive biomarker. So my colleagues and other researchers, they are proving that these biomarkers is a non-invasive, not invasive, it's a non-invasive biomarker for many diseases, like not only in cancer, cardiovascular diseases, diabetes, stroke, and etc. I mean, so many diseases. And there are a lot of microRNA has been discovered. Only few microRNA functions are visible. So what is the research question is, how these microRNA are efficiently used to treat the diseases. For example, oral cancer. Oral cancer, we know that advanced stage, that means aggressive type of oral cancer, which is very, very difficult to treat. Even though there are a lot of drugs has been commercialized in the market, but the side effect is, you cannot imagine, the side effect is more. So people are looking for new therapeutic target. So already we know that some of the genes are upregulated in oral cancer. But the question is whether these genes are used as a therapeutic target. So now the people are looking for microRNAs. These microRNA regulate the gene function. So what people are thinking is, maybe if you are able to suppress these microRNA, we can get any kind of cellular signaling silencing or some targets, therapeutic targets, especially to treat this kind of deadliest. I think it's audible, right?
Yes, sir, you're audible. You can speak.
Yeah. So it is necessary to have high efficient delivery method. I mean, what I'm saying is, even though you discover microRNA, you synthesize microRNA, but the delivery system has to be very efficient to target the particular cells. For example, there are so many barriers. One is intracellular barriers and extracellular barriers. So we need to cross these barriers so that the disease, we need to suppress the disease progression. That is the ultimate goal. So what people are thinking is, if you able to isolate this microRNA or synthesize this microRNA and then coupled with nanoparticles, because as we know that nowadays nanoparticle delivery is very important, a very important study. I mean, the studies is going on. So the nanoparticles are very efficient to deliver your product. Say for example, microRNA is a product. So we need to deliver in the particular site. So you tag with the microRNA plus your nanoparticle. What will happen is this nanoparticle cross this barrier and it deliver your microRNA to the particular site. That is the things, that is the latest technology is going on, but not in oral cancer, but in other cancer is going on. I think in few more years, it may come to the research side, and then people are using a rat model or mice model to see the efficiency of this nanoparticle and microRNA delivery in the cells. So, as I said, nanoparticle delivery system is working with the clinical level. I mean, in the animal model, but not in the clinical level. But people are trying to use this nanoparticle as a cargo. So you can add your microRNA and then deliver to the particular cells. So as I said, oral malignancy is the sixth most cancer in worldwide, and it's very difficult to treat or manage. The important thing is that the recurrence rate is very high. So even though if you treat, again the patient is done with the tumor. So the mortality rate is also very high in developing, also in the developed world. So what the people are looking at, therapeutic target and a good biomarker. So that should be a sensitive one, and then it should not be a non-specific one. It should be a specific one. So there are a variety of delivery system available. Say for example, exosome. Exosome is is a kind of a cargo. So the exosomes derived from the cells may used as a biomarker. Say for example, exosomes also contain microRNAs. It also contains other RNAs. So people are efficiently isolate these RNAs. For example, you compare with the normal cells and the tumor cells, isolate the exosomes and compare these exosomes. So say for example, if the exosomes is very high and say for exosome microRNA derived exosome is very high, you consider this microRNA as a biomarker. So these are all the research is going on. So there are certain things which is not yet established. Say for example, if you treat or if you isolate the microRNA or you synthesize it and introduce into the cell system, what will be the immune system? So yet to research is going on, and what will be the nanoparticle tagged with the microRNA? How it is influence? How it is provoke the immune system? So these are all certain questions going on, but since research is going on, I can't able to tell the exact truth. So as I said, this microRNA is nothing but in the cells itself. Okay, so it is originated from the nucleus and then transported to the cytoplasm, and there are certain protein is involved. For example, exportin is a protein which exports the microRNA from the nucleus to the cytoplasm. Then the cytoplasm, in the cytoplasm, the microRNA is called mature microRNA. It's a single-stranded microRNA, and it will go and bind to the 3' UTR, that is untranslated region of the gene. You know that there are two genes in the cancer cells. One is called oncogenes and the tumor suppressor genes. We know that oncogenes always upregulated in the cells, cancer cells. Tumor suppressor always downregulated. So what this microRNA is, each and every microRNA has its own therapeutic target. So it will go and bind to the oncogenes or tumor suppressor gene, depends upon the situation or microenvironment of the particular cancer cells. So it may upregulate the disease progressions or suppress. This is the concept of microRNA in the cancer cells. So I hope you understand. So regarding the therapeutic aspect, as I said, there are so many translational promising molecules has been established. Say for example, if it is highly upregulated, the microRNA considered to be the oncomere. We can call oncomere. So we can synthesize the opposite microRNA, which is called antimere. So you can synthesize and introduce into the cells. So this antimere go and bind to your microRNA, and then you can suppress those oncomere, so to protect the cells. In other words, there are microRNA mimics are available. So if it is tumor suppressor, your microRNA act as a tumor suppressor, your mimics will go on induce the expression of the tumor suppressor microRNAs so that you can protect the cells. these are all certain kind of molecules is available, but the complete research is not yet well established. So people are working with cellular models, for example, cell culture models, and then see these microRNA used to are involved in the proliferation, migration, invasion, and apoptosis. In other words, the second thing is they will deliver this microRNA into the animal model and see the expression. So either way, the research is going on, but the complete system is not yet completely established. And some of the microRNAs already proven, but the clinical response or clinical research is undergone. I mean, it's going on. So the choice of microRNA also decides the fate of microRNA in the cells. So we need to choose particular microRNA. For example, if I choose the oncomere, so I need to choose the correct antimere to suppress those microRNAs. These are all the thumb rule if you want to work with the microRNAs. The delivery system is very, very important. Say, for example, there are a lot of companies who are synthesizing antimere. So we need to choose which microRNA we are going to work on and see the cellular signaling pathways. Say, for example, some of the microRNAs are involved in NF-kappa pathway, and some of them are involved in PI3K kinase and AKT pathway. And it depends upon the cellular signaling where you are choosing your target and the microRNA. So these are all some of the options are available. But as I said, we are working with the oral cancer. So what we do is we usually isolate RNA from the oral cancer tissue. So sorry, oral cancer microRNAs. So what we can do is with this microRNA, we are see the expression whether it is highly upregulated or downregulated, depends upon the situations. Then we need to choose the targets, particular target for each and every microRNA, and see the cellular signaling pathways. This is the schematic representation of microRNAs and nanoparticles, how we can use to treat the various diseases. For example, as I said, this microRNA are come out of the nucleus and then mature into the cytoplasm. So if you couple it with the nanoparticles, we can use as a the nanoparticle used as a cargos, and the microRNA used as a therapeutic target. And this is not a complete representation. I'm just giving the idea. And this table represents there are so many cancer and related microRNAs and with the nanoparticles. So people are using gold nanoparticles, silicon nanoparticles, and then they coupled with that and to see the therapeutic aspect in the particular cancer. Say, for example, colorectal or in osteoblastic cells or lung cancer cells, ovarian cancer. So many cancer, people are concentrating each and every microRNA and how it is regulated in the particular cells. So recently, we are working with microRNA 138. So we found that this microRNA is tumor suppressive. So what we did is we isolate RNA, total RNA. From that total RNA, we isolated microRNA. And with the use of qPCR, we can able to see how this microRNAs is regulated. For example, if this microRNA is targeting a lot of genes. Fortunately, these genes are involved in the various cancers. So, for example, ISG15, it's involved in the cell cycle regulations. TGF-beta1, tumor growth and metastasis. And delta NP63 metastasis. YAP1 is cell proliferation and apoptosis. So it revealed, this study revealed that the microRNA 138, it acts as a tumor suppressive, and if it is silent in the cells, what will happen is the target genes are upregulated. So controversially, we may think that we can use microRNA mimics to replace this microRNA 138 and see these target genes how they are regulated in the cells, especially in the oral cancer OSCC cells. So I think our team is working on that. I think soon we will publish something. So the next microRNAs what we studied is microRNA 185, and this is recently published in oral cancer, oral oncology. So we see the highly upregulated microRNA 185 in the oral cancer YCC cells. And as I said, we isolated and then we see the expression using qPCR. The oppositely, I think microRNA 138 act as a tumor suppressor, and microRNA 185 act as a oncomine, that is highly upregulated, highly expressed in the oral cancer OSCC cells. So we have some confusions how this happened. So we are planning to link this microRNA 138 and microRNA 185 in the cellular signaling pathways, whether some other novel genes or key genes is a target for these microRNAs. We are seeing that, and we are trying to prove it in an in silico system. I think soon we will going to publish that. So the surprising thing is that these two microRNA act oppositely in the oral cancer cells. That is the discovery of our team. I think this research is going on now. So, yeah. So thank you for your patience. If there is any questions, please let me know.
Thank you, sir. Thank you for your insightful talk.
I
So,
think everyone is understanding. I think a little bit fast. I hope you understand.
yeah, sure. So, there are any doubts you can ask Professor Durairaj Sekar?